I will start by introducing the company. Most of the day we'll focus on ulcerative colitis and cobitolimod and the conduct study. So I will try to cover everything else about the company here in a couple of minutes. Of course, we have our standard forward-looking statement that we try to estimate what will happen and predict the future, but we are not sure that everything we predict will actually happen in the end. So briefly, InDex Pharmaceuticals is a drug development company. We are based here in Stockholm on the campus of the Karolinska Institute, which is also where the research once originated that we're working on. We focus on immunological diseases with a high unmet medical need, and our foremost drug candidate is called cobitolimod that we are developing for ulcerative colitis. And we've come a long way with this asset. We're in late-stage clinical development. We're currently running a fairly large phase 2b dose optimization study across Europe. It's called Conduct, and we'll hear a lot about that. The strategy of the company is to partner cobitolimod prior to phase 3. I will talk a bit about our business development efforts, how we will make that happen. Apart from cobitolimod, we have a broad portfolio of chemically similar substances. Cobitolimod and these DIMS compounds are short snippets of DNA that we can use to modulate the immune system via the toll-like receptor 9. DIMS stands for DNA-based immunomodulatory sequences, to illustrate that these are short sequences of single-stranded DNA. They have a potential in a variety of inflammatory diseases that they have an anti-inflammatory effect. We went public in October 2016. We had an IPO and raised 250 million Swedish crowns. That's about 25 million euros, primarily to finance the Conduct study. And since then, we are listed on the Nasdaq First North in Stockholm. Our ticker is InDex. Our main shareholders are actually the same that we had when we were private. We have SEB Venture Capital, so the Swedish bank SEB, their proprietary venture arm. They have 23% of the company. We have the Swedish Industrial Fund, or Industrifonden, with 21% of the company. And then Swiss-Norwegian life science VC Neomed Management, who holds 11% of the company. I will talk a bit about the people behind the company, the ones some of you will meet today and some that you won't. If we start with the board of directors, we have a very senior group of people with proven track records from the industry. If we start with our chairwoman, Dr. Wenche Rolfsen, she had a long, successful career at Pharmacia and Quintiles in clinical development or drug development. And she's now the chairman not only of InDex, but also BioArctic. She's a board member of Swedish Match and Recipharm. She has a lot of experience from listed companies and larger ones. Then we have Dr. Uli Haxell. He's originally a professor in organic chemistry from Uppsala University. And then he had a successful career at Astra before he moved to California more than 15 years ago and took the helm at Acadia, which at the time was a small startup. And then he, over 15 years, built that to a multi-billion-dollar company on the Nasdaq. We have Stig Løkke Pedersen in the back with the glasses. He was the chief commercial officer at Lundbeck, the Danish CNS company, for more than 10 years, and was part of the Cipralex story there, building the company to an international player. He's now the chairman of New Evolution and other First North listed companies and other companies he's involved with. Then from SEB Venture Capital, we have Andreas Penneval. He's actually here today, in the back there. And from Industrifonden, we have Lennart Hansson, who used to work at Astra and was the CEO of Orexis, but then he's been investing in life science companies for a long time. I don't see Lennart here yet. Oh, there he is, in the far back. The management team, I think we have a very experienced management team, a lot of industry experience. I'm the CEO of InDex now for 3 years. Before that, I was more than 8 years at Diamyd Medical, of which a bit more than 4 as the CEO. Diamyd Medical was at the time listed on the big board in Stockholm, but a similar small drug development company. We have Pernilla Sandvall, who you will hear later. She's our Chief Operating Officer. She spent more than 20 years at Merck MSD in clinical operations. So we're very happy to have her experience when we now running the CONDUCT study. We have Dr. Thomas Knittel, Chief Medical Officer. He's a professor of gastroenterology in Germany. And he's been working for both small and large pharma companies, like Novo Nordisk, for example, in both commercial and research roles. And then we have Johan Gilius, our Chief Financial Officer. He will not speak today, but he's here. If you wave, he's in the middle there. Johan spent more than 25 years at Deloitte and left as partner a couple of years ago and joined InDex about a year ago, pretty exactly. We are supported, or we support, depending on how you view management's role, by a very dedicated and engaged team of 17 people. We have 7 employees and 10 fixed consultants with various expertises within pharmaceutical development. And we work with an outsourcing model, so we outsource the actual experiments or trials or manufacturing to contract research organizations or contract manufacturing organizations, which makes for a very flexible and cost-efficient organization that we don't build our own factory, but we actually rent the factory when we need one. To support that on the science side, we have a scientific advisory board, and we have a very prominent panel of key opinion leaders, among which both Professor Reinisch and Professor Atreya are part, that help us with designing clinical studies and evaluating our results and helping us to find a way forward. As I said, we have this portfolio of more than 150 DIMS, DNA-based immunomodulatory sequences. So it's a very broad portfolio of new TLR9 agonists with different characteristics regarding their immune modulatory effects, which gives them potential in inflammatory disease. The only one in the clinic is cobitolimod, so it's much further advanced than the others, but if we have a success with cobitolimod, that will validate the whole platform. So we're really looking to use this portfolio to broaden the development pipeline. So we are currently running a preclinical development project with a couple of DIMS in order to diversify the pipeline, and we're looking in inflammatory diseases outside of ulcerative colitis and inflammatory bowel disease. So we won't compete with our own cobitolimod. This will allow us to leverage the historical investment which has been made in constructing this portfolio and building the technology platform. We can leverage the pharmaceutical development experience, expertise, and knowledge that we've gained from the development of cobitolimod. And we actually got a grant from the Swedish Innovation Agency for this work in 2016. We got almost 2 million Swedish crowns, and that grant runs until the end of this year. So hopefully we will be able to report some good results by then. I'm very happy to tell you that you can get the printed annual report outside. It was actually published online yesterday, so it's been printed overnight. So it's brand new. I haven't even had time to flip it through myself. So it's a very fresh production where you can learn a lot about ulcerative colitis and cobitolimod and the company. But of course, you can also see the numbers for last year. Regarding revenues, as most companies in our situations, we don't have any meaningful revenues. We spent about 70 million Swedish, so we made a 70 million loss last year, which is not strange for a company like ours either. And probably the most important number here is how much cash do you have in the bank? That's usually what people ask. We had 125 million at the end of 2017. When we did the IPO, we raised 250 million gross, but we only got 200 net because we had some bridge loans, etc., that we had to pay off. So we have about half of the money we raised left. The assessment is that this will see us through the main results of the conduct study, where we expect results at the end of this year, fourth quarter 2018. Which is also what we said in the prospectus for the IPO. So we're according to plan regarding the study and the finances. So the strengths of InDex in summary. We see that there is a large commercial potential in cobitolimod. Ulcerative colitis is a debilitating disease with high unmet medical need. We have annual sales of biologics in ulcerative colitis that amount to more than $5 billion per year. And we'll hear a lot about their pros and cons shortly. So there are quite a few problems with the current treatments, which is why we see cobitolimod has a high market potential as a safer, more efficacious alternative to the currently approved drugs with a novel mechanism of action. As I said, we come a long way. We're in late-stage clinical development. We have extensive clinical experience where we have had an excellent safety profile so far with cobitolimod. And now we expect the main results from the phase 2b dose optimization study towards the end of this year. The study is designed to potentially provide substantially higher efficacy than we've seen before, than the current approved products and also the current industry pipeline. That would make it a very attractive asset for potential partners. As I said, we're looking to partner this prior to phase 3. And if we are successful, cobitolimod will validate also the other DIMs, so the whole portfolio with potential in inflammation would become more interesting. And again, we have an experienced board and management supported by a very dedicated professional workforce that are running this. So that's what I had to say on this, and I will invite Professor Reinisch to the stage, and we are very happy and honored to have Professor Reinisch here today to share his expertise in inflammatory bowel disease, in ulcerative colitis, and in the treatment of the therapeutic landscape. Let me just open your slides as well, see if they show up here. So, Professor Reinisch is directing the IBD study group at the Medical University of Vienna in Austria. He's been managing patients with IBD since 1992, both from the clinical routine perspective and from a clinical research perspective. He's been very active in on a European level. He's a co-founder of the European Crohn's and Colitis Organization, and he's an honorary member of ECHO. He's an expert in designing, conducting, and interpreting the results of clinical trials in inflammatory bowel disease and ulcerative colitis. He's been an investigator in almost 100 studies. And we are very pleased to have him as a key opinion leader on the KOL panel of InDex, and as a medical advisor in the conduct study, where he is one of the central readers of endoscopy images. So you really get to know the patients of this study. So please.
Thank you. So thank you, Peter, for this kind introduction and the kind invitation here to Stockholm to this important event for InDex. And I'm also grateful for this long-time collaboration, which was really interesting and hopefully very successful soon as well. So you indicated it already before. Ulcerative colitis is a debilitating disease with a high unmet medical need, and I would like to delve deeper into this topic and give you some insights into this. So first of all, I think it's important to remind you that ulcerative colitis is one of those two major phenotypes of inflammatory bowel diseases. Ulcerative colitis is one, the other one is Crohn's disease. And they are differentiated by certain kind of characteristics. So ulcerative colitis is much more seen as a continuous disease which involves only the colon and the rectum, which affects mostly the more superficial layers of the mucosa, the mucosal and submucosal layers, which has a certain kind of type of immunological response, which is defined as a Th2 response, and it has a very strange and odd relationship or association with smoking, because it's one of the few diseases which improve under smoking, and where smoking seems to be protective. And I had some patients who stopped smoking and actually they deteriorated, and I advised them to start smoking again, and so we could avoid colectomy, which is the removal of the colon, and that's true stories. Based on the fact that this is a chronic inflammatory process, chronic inflammation can always induce cancer, and therefore we also see in those patients an increased risk of colorectal carcinoma. And it's not only a disease which is limited to the colon, it's also a disease which can show up with manifestations outside of the intestinal tract by involving the joints or the skin or the eyes. Whereas Crohn's disease, it's much more a patchy disease. It can involve the entire GI tract. It can involve all layers of the GI wall, and smoking is a risk factor and deteriorates usually the disease. Nowadays, delving into the pathogenesis of ulcerative colitis and Crohn's disease, it's getting quite complicated, and Professor Atreya will share you more insights. I would only like to tell you that the disease is occurring at the interface of the intestinal microbiota, the epithelial barrier where the TLR9 is expressed on the cells, and the intestinal mucosal system, which is an intricate system where the signal from the mucosa usually elicits some anti-inflammatory impulses and tolerance impulses. But in the disease state of ulcerative colitis, the entire immune system is ramped up, and an increased response of T cells and specified T cells like Th2 cells and Th17 cells is, so to speak, engaged to elicit inflammation. And what we also know from our patients with inflammatory bowel diseases, including patients with ulcerative colitis and Crohn's disease, that there is an imbalance of the intestinal flora. So we, as human beings, have more bacteria in our intestinal system than our own cells. But we also know that the abundance and the richness and the composition of this intestinal flora in patients with ulcerative colitis is disturbed and reduced. And we do believe that this is a major impulse for the chronicity of the inflammation. So we can differentiate different types of ulcerative colitis. As I stated before, usually it starts as a disease in the rectum, which then extends proximally to the left-sided colon, and then in its highest extent it can reach beyond this splenic flexure or left flexure and affect the entire colon. And therefore we're talking about proctitis if it's limited to the rectum, left-sided colitis if it's limited to the left colon, or more extensive, or pancolitis if the entire colon is involved. And dependent upon how extensive the disease is, the patients are suffering from different types of symptoms, but the key symptoms as such are the addition of blood in the stool, so bloody diarrhea, and another important symptom as such is the more of the mucosa is involved, the more likely it is that there is a higher rate of bowel movements, leading in the end to diarrhea of these patients. So far on the level of symptoms, how does the disease look like on the level of endoscopy? So this would be the normal situation, a perfectly normal mucosa or a perfectly healed mucosa in a patient with ulcerative colitis. And from this on, we can differentiate different types of severity on the level of the mucosa as visualized by endoscopy. So a mild stage would mean that this normal mucosa has disappeared, and there is an erythema, and this vascular pattern you might see here has disappeared. In the moderate stage, the first lesions are coming up, erosions, which are tiny ulcerations. The mucosa is friable, which means if you touch the mucosa, even only lightly, then it starts to bleed. And the full blown picture of severe disease is a disease where you see ulcerations and spontaneous bleeding with the endoscope. And ulcerative colitis is not only a disease which has rectal bleeding and an increased stool frequency as symptoms. It also has other symptoms which are very restrictive for the social and professional life of our patients with ulcerative colitis. Symptoms like incontinence, symptoms like tenesmus and urgency. So the immediate urgency and the immediate need to see a restroom, and this limits the patients in their social and professional life hugely. What about the causes of ulcerative colitis and Crohn's disease? Ulcerative colitis and Crohn's disease share some types of genetic susceptibilities, and ulcerative colitis as such nowadays has also realized as a disease which has a number of genetic susceptibility genes attached to it, but it's still not clear on how this difference and this heterogeneity of the genetic composition of the DNA, how this translates into different kind of phenotypes and disease patterns of our patients. And as I mentioned before, usually ulcerative colitis is considered as a continuous disease, but nowadays we also see discontinuous forms of the disease forms where the rectum is not involved, where we see apart from the rectum other involvements in the colon, where we even can see involvements of the small bowel and the duodenum, or involvements of the bile tract. We see differences in the disease extent, in the disease severity, in the age at onset, and nowadays we see more and more young patients and pediatric patients showing up with ulcerative colitis. We see differences in the disease course, differences in the serological composition and in serological markers, and of course also differences in the response to treatment. And this is just one of those examples from a Scandinavian study on ulcerative colitis, an inception cohort where the course of the disease was prospectively investigated over 10 years. And what is important here, there are some patients who present up front with a moderate to severe maybe course of the disease, but then over the years that the disease seems to improve dramatically. But what we usually see in clinical practice are those cases who are either chronically, continuously active, which makes 6% of the cases, or those cases which make about 37% of all cases, where the patients have a disease course where phases of flares of deterioration are interchanging with phases of clinical remission. Nevertheless, even despite that our treatment strategies are improving over years, we still see 10% of our patients after 10 years of disease in the need of undergoing colectomy, so which means that the colon has to be removed, and that either a stoma has to be applied or a pouch has to be applied, so that the patients can still defecate via a natural way. In pediatric, you see the situation is even worse. So after 5 years from diagnosis, up to 20% of patients need to undergo colectomy. And colectomy is definitely not a cure of those patients, because colectomy is associated with complications. It is associated with increased mortality. It's associated with other complications, with obstruction of the small bowel, with increased numbers of stools leading to fecal incontinence. I talked about the pouch, where the small bowel tissue is morphed into a rectum-like pouch, which is then annealed to the anus. But this pouch can also inflame in 50% of the cases and can develop fistula, impotence, reduced female fertility. These are typical complications of colectomy. And even despite the fact that mortality of our patients with ulcerative colitis and Crohn's disease has normalized in most age ranges, in the age range between 0 and 19 years, still there is an increased risk of mortality of our patients with ulcerative colitis and Crohn's disease as compared to a control group. So what are the treatment targets now for our patients with ulcerative colitis? And I was talking about symptoms like rectal bleeding and an increased stool frequency. So first of all, we would like to get the patients rid of the symptoms. But we also know that there is a disconnect between symptoms and the endoscopic picture, and that sometimes patients could be symptom-free, but there could still be an inflammation on the level of endoscopy. And therefore, we are defining our treatment targets on 2 levels: on the level of the symptoms and on the level of the endoscopy, that the patients need to be free of ulceration, in best case have a completely normal mucosa. And nowadays, when you're reading about clinical trials and the endpoints, you could easily get lost in all those different endpoints which have been brought forward. And this slide should highlight this fact. But nevertheless, more recently, FDA and EMA came up with quite clear guidance on how the endpoints need to be composed, and it's still the resolution of symptoms, particularly the rectal bleeding score and the stool frequency score, and a normal and mucosal healing as defined by the absence of ulcerations on the level of endoscopy. So what kind of treatments now do we have in place to achieve those goals? And this is the current treatment algorithm as it looks like. So if the patient starts with mild disease, usually we are applying 5-aminosalicylates, so an anti-inflammatory compound. This is applied both orally and topically as an enema. So patients are used to apply enema as a standard treatment for ulcerative colitis. If the patients are failing with the 5-aminosalicylates, then the patients are actually moving from the mild disease arena into the moderate to the severe disease arena, and they need to also to ramp up the treatment to immunosuppressives, including oral steroids, including immunosuppressants like thiopurines, azathioprine, 6-mercaptopurine, and the addition of biologics. And currently we have as biologics anti-TNF-alpha agents in place, so compounds which are blocking specifically a cytokine which is called tumor necrosis factor alpha. And for this we have an array of different compounds in place: infliximab, adalimumab, and GL stands for golimumab. And for a few years, we also have a new mode of action in place: anti-adhesion molecules, an antibody which blocks a certain kind of surface marker, alpha 4 beta 7, which is necessary for the T cells and for the inflammatory cells to get recruited into the inflamed mucosa, and by blocking this interaction, the recruitment into the inflamed mucosa is stopped. And in the worst case scenario, patients have a fulminant course of the disease. They need to be hospitalized. They need to receive intravenous steroids. If they don't work, they need to receive immunosuppressants intravenously, including cyclosporin A or tacrolimus or infliximab. But even despite all these different treatments we have available, there's still an unmet need with regard to the outcomes. So the outcomes currently are still not optimal. So the 1-year clinical remission rate we can achieve with the existing drugs like adalimumab, infliximab, dependent upon the design of the study, ranges between 16 and 40%. So which means 60% of the patients don't achieve the goals as we define it. And these goals are, let's say, much more the more simple to achieve goals. So you can even increase the bar of your goal and say the patient need to be steroid free or need to have long-term remission. Then those rates would be lower. You could even say mucosal healing is not enough, so the absence of ulcerations is not enough. The mucosa needs to look completely normal. This would be also a higher goal, which is even more difficult to reach. We also know that patients who have been exposed to previous treatments, particularly to biologics, that they are responding by far worse as patients who are naive to those treatments. And nowadays also the agencies, FDA particularly, is coming in and also would like to increase the bar of the endpoints, not only achieving the goals on the level of the endoscopy. They would like to see histologic remission, so meaning if the histopathologist is looking at the slides of biopsies, then they would like to see some signs of remission also on that level. We also take biomarkers to measure the inflammatory load of our patients with ulcerative colitis. And nowadays there are intentions to get the patients even free of inflammation on the level of biomarkers. And of course there are safety issues. And the safety issues, they are attached with most of the biologics. They are particularly with the anti-TNF alpha agents, where there is an increased risk of infection that has been shown repetitively with the immunomodulators, where there is even an increased risk of cancer, particularly lymphoma and non-melanoma skin cancer. So what can we do nowadays to optimize our treatment in patients with ulcerative colitis? And we have different types of strategies. We can drive the optimization by following up the patients on their symptoms, and if the patients don't improve their symptoms, then let's increase doses or change the treatment. Nowadays we also have strategies where we tight monitor these symptoms, even by measures of telemedicine. But what is becoming more and more important is not only the tight monitoring of the symptoms, because I explained to you, symptoms not necessarily are reflective of the inflammatory load. Therefore, we tight monitor the inflammatory biomarkers, either in the serum or in the feces. Particularly with the biologics, we can also measure the concentrations of the drug in the blood, which is the therapeutic drug monitoring, and can adjust the treatment according to the serum concentrations of those biologics. We can try to combine treatments, but this is something which is not necessarily far progressed so far in the treatment in UC. And more recently, the first study has started trying to combine the treatment of two biologics. And I do believe that combination treatment it will be an important part part of the future of the treatment of patients with ulcerative colitis. Precision medicine, so can we bring in all the new knowledge from genomics and microbiomics and proteomics and how all this big research application are called to specify which patient might have the best benefit out of a certain drug. This sounds still somehow futuristic for our patients with IBD, even despite this has already reached patients in oncology. So what kind of modes of action do we have in place? And there is a plethora of new modes of action coming in. I mentioned already the anti-cytokines, anti-TNF-alpha agents, which are already available. I mentioned already cell migration, the integrins, particularly alpha-4 beta-7, which is vedolizumab. But as you can see, there are much more anti-integrins now in phase 2 and phase 3 studies, and most likely the data will become available over the next few years. There is also a modulation of sphingosine phosphate receptors, and also we have here the first data from phase 2. Then there are these anti-inflammatory principles with small molecule JAK inhibitors, which are inhibiting signal transduction cascades in inflammatory cells. PDE4 inhibitors, which also works anti-inflammatory, and the TLR9 agonist with copitolimod. And we also would like to enhance the barrier between the microbiota and the intestinal immune system with microbiota modulation or modes of action like the application of phosphatidylcholine. And I give you only a few examples of more recent trials. So this is data with tofacitinib, a pan-JAK inhibitor, which blocks the signal transduction cascade of JAKs in inflammatory cell types. And this is one of the breakthroughs in the treatment of UC, but you can see already the limitation of those breakthroughs. This is Octave 1 and Octave 2 induction studies at week 8, and those patients who have responded during induction then moved on in a re-randomized fashion to Octave sustain. And what you can see after 8 weeks, a clinical remission rate 18.5% in Octave 1 with tofa 10 milligrams and 16.6 with tofa 10 milligrams in Octave 2. So clinical remission. This is the limitation, which also tells you about 80% of the patients were not in remission. So there is a high unmet need, and with another molecule like osanimab from phase 2, we observed very similar remission rate, a delta of about 10%, 10 to 12%, but still quite a limited number who have achieved this endpoint. This is another compound, apremilast PDE4 inhibitor, which results were shown more recently at the European Crohn's and Colitis Organization Congress in February. Another study which is positive and we believe could be another breakthrough. But again, the limitation is quite obvious. We have a relatively high placebo remission rate with 13%. We have with 30 milligrams BID of apremilast 31% of remission, but there is no remission in dose relationship, because with the higher dose there is no efficacy observed versus placebo. So the phase 3 program will definitely show how successful this drug could be. So how might then could the future treatment algorithm of ulcerative colitis look like with the advent of the biosimilars, so the cheap versions of the biologics? I think most of the new modes of action will be placed in the moderate to severe category of patients treated with ulcerative colitis, and most likely behind the biosimilars, maybe apremilast, which has a which seems to have a nice safety profile, might have the potential to be positioned earlier, maybe in those patients who have failed 5-aminosalicylates. But capitolimod might have the potential to be placed in both categories due to its safety profile, and you will see more afterwards. It could be placed early on in the treatment of patients, but might have also the potential to be placed in the moderate to severe category. So I'd like to conclude at this point by summarizing that there is a high unmet need in the management of patients with ulcerative colitis on various dimensions. First of all, in the understanding of the biology and the heterogeneity of the disease. Secondly, by the way on how we define the endpoints. So even the agencies are not quite clear what the best endpoints in clinical studies look like. By the treatment optimization strategies in clinical practice are still missing. Something is ongoing here in Crohn's disease, but still missing in ulcerative colitis. And I think there is a dire need of new modes of action. Thank you very much.
Thank you very much, Professor Reinisch. Introduction to IBD. All right. Hello. Okay. Thank you again, Professor Reinisch, for that introduction to the disease of ulcerative colitis and the current treatments. I will just shift the slides here before I welcome Professor Atreya to the stage. Very happy to have you here to introduce cobitolimod, our lead drug candidate at InDex. Since 2016, Dr. Atreya is the Heisenberg Professor for Translational Immunology in IBD at the University Hospital in Erlangen in Germany. For the non-Germans, I should explain that the Heisenberg Professorship is one of the most prestigious professorships in Germany that is awarded by the German Research Association to outstanding young scientists. In 2015, Dr. Atreya also received the Rising Star Award from the United European Gastroenterology Federation. So it's an up-and-coming man. We are pleased to have him on the InDex KOL panel and as the principal investigator in our CONDUCT study. So please, Raya.
So, Peter, first of all, thank you very much for these very kind words. I would have liked to listen on and on to these, and to be called a young scientist in this beautiful morning in Stockholm is also a good sign. So I'd like to talk about cobitolimod. So as you can see here, a promising first-in-class drug candidate. I'd like to introduce you certainly to the mode of action, why this is a new drug substance class that has been opened, and what are the data regarding mode of action and of the last clinical trial that was conducted. So you can see here, cobitolimod is indeed a first-in-class compound. It is a DNA-based synthetic oligonucleotide that sort of really represents a new substance class that might enter the treatment of inflammatory bowel disease patients. So you can see here cobitolimod, and this has a certain motif, a motif of denucleotides that really mimic microbial DNA. So then, if this cobitolimod then binds to a receptor that is also able to recognize bacterial DNA, due to the motifs that resemble bacterial DNA, this receptor is activated. So for instance, TLR9, so the toll-like receptor 9, is one of the receptors that are able to recognize bacterial DNA. And by cobitolimod sharing these motifs with bacterial DNA, the receptor is then activated. So this is how the body really is able to deal with bacteria, because by recognizing bacteria, the modulation of the mucosal immune system on a local level is done. So this leads to the production or to the elevation of anti-inflammatory effects on the mucosal level, then leading to the healing of the colonic mucosa. So by this concept, or this concept was then used also, of course, for cobitolimod being a representative for very, I think, promising treatment option in patients with ulcerative colitis. So we have talked about this TLR9, the toll-like receptor 9. I'd like to introduce you a little bit What is the toll-like receptor 9? So it is a key pattern recognition receptor of the innate immune system. So all of us really have this toll-like receptor 9. And it's really expressed on intestinal immune cells and on the surface of epithelial cells, and has been shown that the expression level of toll-like receptor 9 really correlates very well with the level of inflammation in ulcerative colitis patients. So as I've said before, this toll-like receptor 9 now recognizes so-called unmethylated CpG sequences. So these are dinucleotides that are also part of the bacterial and viral DNA. Now this corbitolimod, as a drug substance, contains these unmethylated CpG motifs, so like bacterial and viral DNA also do, and they mimic this bacterial DNA and therefore represent a natural ligand of the toll-like receptor 9, and is then similarly able to really activate this local immunomodulatory effects. So then the binding of corbitolimod to this toll-like receptor 9 really aims to restore the disturbed innate immune function and balance in immune cells, which is really existent in patients with ulcerative colitis. So now we look at corbitolimod as this drug substance, and you can see here, yes, indeed, it has a certain history of development. So some trials have been performed, some studies. You can see with a varying number of patients, and also, of course, for dose finding, different dosings were also used. And then all in all, roughly 250 patients were treated with corbitolimod across all these studies, which of course is a very good basis looking at efficacy and even more of safety of this compound. So I'd like to introduce you now to the results of the biggest study that has been conducted so far, the so-called COLLECT study, a randomized placebo-controlled trial. And you can see here, so this was the basic design of the so-called COLLECT study. So all in all, 162 patients were screened, and then 131 patients were randomized. And now who were these ulcerative colitis patients that met the inclusion criteria? So all these patients had moderate to severe active ulcerative colitis. And you have heard with Professor Reinisch, what does it mean to have active ulcerative colitis? So on the one hand, they had clinical activity. So this is also reflected by the so-called chi-score, equaling or above 9. So this means patients really had clinical symptoms, as you have seen in the presentation before, with high stool frequency, for instance, and rectal bleeding. Moreover, it was not sufficient that they only had clinical symptoms, they also had to have mucosal inflammation on the endoscopic level. So all patients also received endoscopy at baseline, and they had to have a Mayo score equaling or above 2, meaning they had at least had to have ulcerations into mucosa. So this really shows you, yes, they had clinical and endoscopic symptoms, and moreover, to make it even harder as an inclusion criteria, they had to be refractory to available therapies. So this shows you, yes, these are patients with a certain level of inflammation. So now these 131 patients were then randomized 2-to-1, and the one treatment arm was the continuation of the standard of care, and additionally corbitolimod application was done, and the other treatment arm was the continuation of standard of care treatment, and the placebo was added. Now how was this drug substance added? You can see here the dosing of corbitolimod with 30 milligrams took place at week 0 and week 4, and then the primary endpoint for the assessment of the effectiveness of this drug substance was done at week 12. It was a novel mode of application also in the study regarding corbitolimod, or the drug substance, because an endoscopy was done in the patients, and then the drug substance was applied onto the mucosa locally via a spraying catheter during endoscopy proximal to the site of inflammation. And now if a patient had really inflammation throughout the whole colon, so-called pancolitis, the drug substance was applied in the transverse colon, which means it was the middle of the colon. So it was a novel mode of application, but which of course ensured really that the drug substance reached its target, which was the mucosa, or the inflamed mucosa of the ulcerative colitis patients. And you can see here then at week 0 and 4, the drug substance was applied. So who were these patients? Some details regarding the included patients. You can see here on the left-hand side the placebo arm, on the right-hand side the corbitolimod arm, and then the overall arm. So these patients were roughly around 40. There were a certain percentage of smokers. Then I think, which is of course very important, is the duration of disease of ulcerative colitis. you can see really that all these patients were suffering over 9 years in the mean of ulcerative colitis, which really shows, of course, that this disease already had an effect on the mucosa. It was sort of remodulated. Then they all had a clinical activity score, which I said before. It was all equaling across all treatment groups with 11. Patients also had to have endoscopic activity. We have seen that before. This is ranged from 0 to 3, and patients had a mean of 2.6. So this again shows you, yes, they had ulcerations and was quite severe disease. And also, we have heard that by Professor Reinisch, new treatment outcome scores that we are looking at on a histological level. The so-called Gebow score was used. So this is a score that goes from 0 to 5, 5 being the highest activity. And here again, a mean of 3.6 was achieved by all treatment groups, which I think again underlines the severity of inflammation in the treated patients. Then, which is important, we saw before they had to be refractory to available therapies. So all these patients, roughly 80% of all these patients were refractory or were exposed unsuccessfully to immunomodulator treatment. And then you can see here roughly 40% of the patients were also anti-TNF antibody treatment experienced. So you can see here again, yes, these were patients with severe inflammation being refractory to either immunomodulatory or anti-TNF therapy. Then we look now at the primary endpoint, which of course is very important for each study. So as I said before, drug application took place at week 0 and week 4, and the primary endpoint was defined as week 12 as clinical remission. So first of all, if you look at the cobitolimod line, this is the line in blue, you would see that yes, over 40% of the patients reached this very hard endpoint of clinical remission, which at the beginning you would say this is a very good trial result. 40% of patients reaching clinical remission. This is really even better than probably all the therapies that we have seen before. But if you look at the orange line, this is the line of placebo treatment, you can see the same amount of patients also reached clinical remission in the placebo arm, which of course makes you wonder a little bit, because these are patients with severe disease treated with a placebo, so how can they reach 40% of remission? And this was also, of course, then the basis to really look at the data more stringently, and of course to look at the endpoint defining clinical remission even harder. So as you can see here now on the left hand side, so which are the now recommended endpoints that have to be used in clinical trials or really define if a patient responds to therapy or not. And we have also again heard this by Professor Reinisch. So once it is stool frequency and rectal bleeding. So these are of course the patient reported outcome measures which really define how a patient suffers, how many times he has to go regarding stool and how severe is the rectal bleeding. And then of course these two compounds are then also added by endoscopic level of inflammation. So these three outcome measures are really the ones also recommended by the FDA and the EMA nowadays that really define treatment response. And now if you look at the score that was used in this trial on the left hand side, you can see here it's depicted by COBIT, so cobitolimod. So in the trial this was the one that was used, the so-called chi-score. You can see here that on the left hand side, stool frequency, rectal bleeding are included, but endoscopy is really not assessed in this endpoint defining clinical remission. And there are endpoints that really nowadays do not really play any role in defining treatment response, like physician assessment. This is a totally subjective score, abdominal cramps, extraintestinal manifestations. So you can really see there's a lot of noise coming from endpoints that are not really regarded as meaningful nowadays in trials with ulcerative colitis patients. On the right hand side, you can see the so-called DIE score. This is the score that has been used recently in the approval of all the drugs that are approved nowadays in ulcerative colitis, like the anti-TNF antibodies infliximab, adalimumab, and vedolizumab. And here you can see that stool frequency, rectal bleeding, and endoscopy are part of this score. And there's also a fourth compound, which is called physician assessment. And this has been now really erased because, again, this is a very subjective outcome measure, not an objective one. So this is also being erased. So again, you can see here then, when comparing what is recommended by the FDA and EMA nowadays, and then what has been used in the COLLECT study with the chi-score, there's really discrepancy because there are a lot of outcome measures that have been included in the score that do not really play a role in defining treatment response. So what are then the explanation for the very high placebo rate that was seen in the study? So that also explains why cobitolimod clearly missed the primary endpoint at week 12. So first of all, there was no really hard definition of a primary endpoint. So the chi-score equaling or below 4, you could see again, it includes rectal bleeding and stool frequency, but also a lot of other noisy outcome measures that can, of course, then disturb the picture that you see. So that was not really a robust scoring system for ulcerative colitis disease activity. And it's known also to induce high placebo rates by subjective outcome measures. And of course, if you think the drug substance was applied at week 0 once and also at week 4, then looking 8 weeks after the last application at week 12 probably is a too long time to look at a primary endpoint, which of course then also drives a high placebo rate also due to the continuation of concomitant medication, for instance glucocorticosteroids. So then we look at predefined secondary outcome measures in this clinical trial and also look at these endpoints that are also used now in clinical trials and are recommended by the FDA and EMA. So first of all is the patient-reported remission. So this is a score that means absence of rectal bleeding. I think this is always something as a physician that we try to achieve, because absence of rectal bleeding is really an outcome measure that has to be achieved by a successful therapy in ulcerative colitis patients, and normalization of the stool frequency in the score. And if you take this secondary endpoint, a patient-reported remission, you can see in blue is the cobitolimod treatment group. You can see a delta of 18% in comparison to placebo, which was statistically significant. And if you now take the third outcome measure that is regarded as very important, mucosal healing, so the absence of ulcerations in patients treated by cobitolimod or placebo, you can again see that there was a delta of 16% in favor of cobitolimod in comparison to placebo, which just missed statistical significance. Now, if you then try to combine these outcome measures, which is the recommendation by EMA and FDA nowadays, patient-reported remission plus mucosal healing, so a very hard outcome measure, you can again see a delta of 18% in favor of cobitolimod in comparison to placebo, which was also statistically significant. And you can also see that this is a very valid outcome measure, because in the placebo-treated patients where you would not expect any treatment effect, just about 2 or 3% of the patients reach this endpoint. And again, I think you have to keep in mind that this endpoint was really investigated at week 4, so just one application of the drug substance took place. So then we look at other predefined secondary outcome measures of the study, and we have seen that these outcome measures are really are of growing importance in the field of ulcerative colitis. So we're looking here at the histopathological disease activity score, again at week 4. So patients underwent endoscopy at week 4 again, so 4 weeks after drug substance application. So I think there are some discussions, what is the best histological endpoints, but if you took a Gibbo score equaling or below 1, so the Gibbo score, the histological disease activity score, goes from 0 to 5, so equaling below 1 is a very hard endpoint. And here again you can see a delta of 11% in favor of the cobitolimod treated patients in blue. And if you take a little milder endpoint, Gibbo score equaling or below 2, which still means sort of histological response, there was a statistically significant result in favor of the cobitolimod treated patients with a delta of 20%. So again, you can see here, besides clinical efficacy and also endoscopic response, there was also response on the histological level after application of cobitolimod. Then I think which is important, if a patient shows response, what happens to him next? So this just looks at patients with patient-reported remission and mucosal healing, so this very hard endpoint, which is now also the new endpoint in clinical trials. And again, as I've shown you before, at week 4, there was a delta of 18% of the patients in comparison to placebo that reached this endpoint and were treated with cobitolimod. And if you looked at this very hard endpoint, which of the patients reached this endpoint at week 4 and week 12, you can see that yes, 70% of the patients that had reached this endpoint at week 4 also prevailed at week 12, still having the response on the clinical remission at the clinical and endoscopic level, reaching statistical significance. Then what is important, because of course anti-TNF antibodies are an established treatment option, but we know and we have seen this with the data there's a large subgroup of patients that do not respond or lose a response to treatment with anti-TNF antibodies. So what were the results if you stratify the patients regarding experience or regarding exposure to anti-TNF treatment or naivety to this treatment option? So you can see here on the left-hand side, these are patients that had been exposed to anti-TNF antibody therapy before. And you can see here there's a delta in favor of corbitolimod if you compare it with the placebo-treated patients. And on the right-hand side, you can see that the drug was more efficacious regarding the delta in anti-TNF naive patients with a delta of over 20% in favor of corbitolimod treatment. So I think this is very well in line also with study results that we know from other drug substances, like for instance vedolizumab, which is also another substance class, which also looked at the efficacy of its drug substance in patients exposed or non-exposed to anti-TNF therapy. So these deltas are well in line with the results of vedolizumab, for instance, showing higher efficacy in patients that are naive to anti-TNF treatment and a little lower efficacy in patients exposed to anti-TNF treatment. But I think what is important now regarding corbitolimod with this very hard endpoint of patient-reported remission at week 4 is that there's efficacy in both anti-TNF experienced patients and anti-TNF naive patients. Then what is also, I think, a very important outcome measure, we have seen this, what are we and also from a patient's perspective trying to avoid is the rate of colectomy. I think one, of course, has to be cautious in this trial regarding the rate of colectomy, but I'd like to show you just the data because patients were really looked after 1 year in the whole trial. And you can see here at 5 months, 3 of the patients receiving placebo had to undergo colectomy, 1 in the corbitolimod treatment arm, and after 1 year, it was the ratio was 3:2. So again, you can see there was a certain effect also by corbitolimod regarding this very endpoint of the rate of colectomy. I think effectiveness is very important, of course, for a drug, but I think really nowadays what defines a drug and also if we apply it as a physician is how is the safety profile. And this is something, of course, what patients also ask us. They want an effective drug, but they want also a safe drug. And I think here in the COLLECT study, this of course was also an important endpoint that we looked after. You can see here patients regarding adverse events, gastrointestinal adverse events, serious adverse events, deaths and treatment-related adverse events. You can see that there was no statistical significance regarding the patients that were treated with placebo or corbitolimod. So I think with this experience we can really see in this randomized placebo-controlled trial that application was with corbitolimod was very well tolerated by the ulcerative colitis patients. So now I've seen all the results, I would say the most important primary and secondary endpoint results and safety results. And we all summed this up in a manuscript, which was then also submitted to the leading journal in the field of inflammatory bowel diseases, which is the Journal of Crohn's and Colitis. So this is also the official journal of the European Crohn's and Colitis Organization, the ECHO. And it was, yes, luckily, I think, accepted, of course, by the journal. It was published in the end of 2016 in the Journal of Crohn's and Colitis. So I think this again shows you, I would say, the interest of the IBD community in this trial because, yes, we saw it missed its primary endpoint, but they could also see, I think, the significance of the secondary endpoints that really play a big role nowadays and also the new mode of action. I think these are the 2 main reasons why it was accepted in this publication and, of course, also its very convincing safety profile. So now we have seen, yes, there's effectiveness and I think the clinical endpoints and endoscopic endpoints that are really used nowadays. The safety profile is very good. And what we have also looked after in a collaboration with InDex is what is the mode of action of this new drug? Because we have seen this oligonucleotide, a new drug substance class. So I think it's always important to know what is the mode of action because only then you can really learn how does my drug work, what is the drug target, and perhaps even to find a biomarker. So we have done some, over the years, some basic science work and translational research work and really collected all our results and submitted it to the ECHO Congress, so the Congress of the European Crohn's and Colitis Organization, which was held in Vienna in February this year. And this is the biggest IBD Congress worldwide. And I think it's over 6,000 to 7,000 delegates that take part. And I think what was we were proud and very happy about this, the abstract we submitted regarding the mode of action of cobitolimod was selected as one of the top 10 abstracts regarding research out of nearly 1,400 submitted abstracts. So I think, again, like the publication, this really underscores the interest of the IBD community also in the compound. So I think what is important is that there is an imbalance in ulcerative colitis patients between pro-inflammatory and anti-inflammatory factors. So really pro-inflammatory factors dominate over anti-inflammatory factors. So if you think like a balance, there's really a dominance of pro-inflammatory factors. For instance, Th17 cells that dominate over anti-inflammatory cells or factors like regulatory T cells. So then we looked, how does cobitolimod work? And you can see here on the left-hand side, this is an experimental colitis model in mice where cobitolimod was applied. This is the blue line, and placebo, the orange line. And this is like a disease activity score. And you can see here, also again, there was a 2-time application of placebo or cobitolimod at week 4 and 8, that the disease activity score was really suppressed by cobitolimod treatment in comparison to the placebo-treated experimental colitis mice in orange. And if we then looked what is the mode of action, we could see that pro-inflammatory cells like the Th17 cells were suppressed by cobitolimod treatment. These are the blue bars, while placebo-treated animals did not show the suppression of the pro-inflammatory cells that really drive inflammation, mucosal inflammation. And on the other hand, on the lower right-hand corner, you can see that anti-inflammatory cells like T cells that express IL-10 were really elevated in cobitolimod-treated mice, while placebo-treated mice really had much less of these anti-inflammatory cells. So we then also looked at intestinal cells that we isolated from mucosal biopsies of ulcerative colitis patients. So if you do an endoscopy, you can take mucosal biopsies of these patients, isolate cells, and then treat them in vitro in a culture system. And again, you can see here, if we added cobitolimod to our culture system, so that's the blue bars, you can again see that pro-inflammatory cells like IL-17A, IL-17F, and IL-6 are suppressed, while anti-inflammatory cells like anti-inflammatory cytokines like IL-10 are really elevated. So again, you can see that cobitolimod is able to somehow suppress pro-inflammatory cells and cytokines and really elevate anti-inflammatory cells and cytokines. And really, as a last proof of concept of our research results, we also looked at biopsies from this COLLECT study. So as you have seen, patients underwent endoscopy at week 0, 4, and optional at week 12. And we took biopsies during these colonoscopies and then stained them for pro and anti-inflammatory cells. And again, you can see here regarding week 4 that patients treated with cobitolimod, there was an elevation of anti-inflammatory cells that express IL-10 or FOXP3 as a marker of regulatory T cells, also anti-inflammatory cells, while the pro-inflammatory cytokine IL-17 on the right-hand side was suppressed by the one-time application of cobitolimod. So then I'd like really to sum up the mode of action. You can see here on the left-hand side, this is how it is in active disease in ulcerative colitis. There's a dominance of pro-inflammatory factors, pro-inflammatory cells that really perpetuate mucosal inflammation in ulcerative colitis, while anti-inflammatory factors and cells are lessened. And now treatment with cobitolimod, I think our research results have nicely shown this, is able really to restore this disturbed balance in ulcerative colitis patients, suppressing suppressing pro-inflammatory cells and factors, and really elevating anti-inflammatory factors and cells, thereby restoring the balance between these two factors and restoring mucosal homeostasis in these patients. So I think we could see really that topical administration of cobitolimod is able to modulate immune response, the endogenous immune response. So we are not inhibiting something, we are helping the patient really to restore his endogenous mucosal immune function, suppressing pro-inflammatory factors and elevating anti-inflammatory factors. So with this, I'd like to sum up all the results. So I think, yes, cobitolimod really represents a potential new medication for patients with moderate to severe ulcerative colitis. We have seen that there's a proof of concept, there's clinical efficacy with a very favorable safety profile. I think we have the experience of treating roughly 250 patients over the years. This is a novel and unique mechanism that is really with a drug substance that is locally applied, so there's no systemic exposure of this drug substance, So this also explains the high efficacy and the good safety profile. And I think in ulcerative colitis, it's important really to look at local treatment options, because then you can get high concentrations of your drug substance at the target site, which is mucosa, which is the inflamed mucosa, and inhibit systemic exposure. And we have seen this with one application. After 4 weeks, you have very good results. So there's also rapid onset of action. And these were the results that I'd like to present you. And yeah, then of course would then like to discuss with you in the next round. Thank you.
Thank you, Professor Treya. Then I forgot to turn on my mic again. One thing I need to remember. Thank you, Professor Treya. You will be invited back on stage shortly together with Professor Reinisch and Dr. Knittel. But I would like to just say that for those following this via the webcast, which I heard was 500 people, we'll also be able to submit questions to my colleague, Charlotte Oddmyre, who is managing the computer here. And her email is on the page where you follow the webcast at the bottom. And you can email her questions to this first panel, but also later on for the final Q&A session where we have all speakers. I should also open this slide here. So for the panel, I would like to invite again all the German-speaking gastroenterologists on stage. And I would like to start with some questions from my side and get the discussion going, and then we can have questions from the audience or from the via email. But I would like to kick off with what for InDex is a very pertinent question to Dr. Reinisch and Treya. So we saw, Dr. Reinisch, you showed all these existing treatments and the treatments under development. Do you really need new drugs in ulcerative colitis?
Yes, I think so for various reasons. First of all, I showed you the limitation of the efficacy of all the new compounds, where particularly during the induction phase, a maximum of 15 to 20% of patients are achieving remission, where the delta to placebo in maximum is around 15%. In the studies, usually these patients are then re-randomized and the responders are then followed up for maintenance, which makes the maintenance profile look by far much better than it usually is. So you always have to take the numbers from the maintenance remitters and need to take into consideration on how many patients have been started the drug up front. So in the end, we end up with 20–25% of patients achieving clinical remission in the end. So that's a fact, and therefore I would believe that a plus of more modes of action is necessary. Another fact is that many modes of action which are currently in place have of course some safety issues. I mentioned an increased risk of infection. I can give the example of tofacitinib, the pan-JAK inhibitor. I showed the data before. There is an increased risk of herpes zoster attached to the drug, which needs to be overcome, maybe by vaccination of the patients up front. There is a signal for an increased risk of non-melanoma skin cancer, for example, and so on and so forth. And so you could find around many of those new modes of actions some issues attached to those compounds. The issue with the biologics is the loss of response over time and the immunogenicity of the drug. So we need definitely modes of action which are not immunogenic, and we need modes of action which are not immunosuppressive, but have much more this resolution potential and the barrier enhancement potential that Professor Treya alluded to with copitolimod.
question for you then, Professor Atreya. What do you see as the advantages of cobitolimod versus other drugs in development?
I think that was also one of the reasons why we looked at the mode of action, because this is really something novel, which we do not have. I think we are using and successfully also using to a certain point antibody treatments that inhibit certain cytokines. But of course, always there's a downside on this, that this really is associated with sometimes severe side effects. So I think, as we have seen here, and probably as our results also show you, the ability to really activate the endogenous potential to restore the disturbed mucosal homeostasis or the disturbed mucosal immune system in the patients is really a big advantage, because nowadays, of course, safety is not only nowadays, but always safety is a very big issue. So I think the local delivery of a drug really to the site where the inflammation is, and that is the mucosa, is really a big advantage, because you get high efficacy due to drug high concentrations, and really there's no systemic exposure, because with all the other drugs we have nowadays, there is systemic exposure, which always really leads to some sort of effects in the immune system, the systemic immune system. So I think with really just pinpointing the effects on the mucosa is a big advantage, really restoring the disbalance between anti and pro-inflammatory factors. So I think with this new mode of action and the associated safety that results out of it, I think this is a big advantage of cobitolimod.
Thank you. Do we have any questions in the audience here in Stockholm to start with?
Yes, in relation to the mode of action, are you really certain that you don't also have an antisense effect, i.e., that you're blocking mRNA?
Maybe I can take that because this is a little bit also about the history of this compound, which was initially developed as an antisense against NF-kappa B. And we have studied that quite extensively, and were not really able to show recently any antisense effect when we study cells in culture. So the best system we can use, we have not really were able to confirm this initial idea about an antisense. I think, of course, you never can exclude it completely. This is there's always a little tiny probability, but when we do control experiments with really antisense compounds which have a clear effect, this was not shown for cobitolimod.
Okay, question?
Excuse me, I'm a shareholder in the company. I'm not an educated person, but I'm curious about when Dr. Atreya says you used a couple of times the word new substance class. What exactly do you mean by that? Is that in efficiency then, that you used it about the competitor and about cobitolimod twice, I think?
Yes, I think this really underlines a little bit, probably I have to say also the excitement, why we also took part in a clinical study, because nowadays, because we are a rather big clinic in Germany, we are approached by several companies to participate in all the studies, which of course we are not able to do because we can only recruit one patient for each study. We cannot recruit him for all the studies. So this is also something that we look upon, why should we participate in a drug study? So for instance, if the fifth anti-TNF antibody approaches us, we'd say, no, what is the meaningful participation? So I think with cobitolimod, the oligonucleotides, I think this is a new substance class, and we can also see there are other substances going that way. And I think with the local delivery, I think this is something that has the charm, I would say, regarding this drugs or substance class, that also is associated with high efficacy rates. I think one has to see that, of course, in the new study also. But I think this is something what we need also, because with the substance classes that we have at the moment, antibodies, we have seen the downsides of it. The JAK inhibitors, there are some positive sides, but also with side effects, something that's already troubling us before they even approved. So I think we need something else, and I think a new drugs substance class is something that might really take this into account. I think that the delicate balance between efficacy and safety.
Maybe when it comes to new substance class, of course, oligos are tested in other indications. So it's not like it's the first ever done in a human. It's a new substance class, a new principle in the field of ulcerative colitis. So no one is really targeting the toll system, which plays an essential role in educating your immune system in the gut, how to deal with, for example, bacteria. And UC is regarded as an overreaction of the immune system. So I think that's the beauty of this approach, that we are targeting a receptor system which is involved in the pathogenesis, not a single cytokine, but a receptor system with compounds new for the field, but also used in other fields.
And I had a question about the positioning in the treatment paradigm. I think you, Dr. Reinisch, talked a little bit about how you could use cobitolimod, but maybe you could expand that a little bit, because I think previously it was thought that cobitolimod could be a good last-line treatment prior to colectomy. How do you see this, and what's your thoughts about that?
I think first of all we would need to see further results then from the next program that Dr. Knittel will refer to. But the first signals that are coming in are showing me that there is a potential in at least two directions. So on the one hand, as Professor Atreya has shown, you have, so to speak, efficacy signals in both patient populations. So a patient population that has been exposed to biologics, but also in the patient population which was naive to biologics. And this is somehow of interest and remarkable, as many of the novel modes of action do show a signal in the naive patient population, but are not necessarily significant, or in the post hoc analysis succeeded to have significant results in a TNF-alpha exposed patient population, like with anti-adhesion molecules. And therefore, you could speculate that you could position the drug at least at two extremes of the treatment algorithm. So one extreme is the one you are alluding to, so in patients who have failed everything, in patients who have failed anti-TNF-alpha agents. But I would also believe that due to the fact that the drug appears to be safe, that the drug appears to be non-immunogenic, and that it's a local delivery system to which patients are used by the 5-amino salicylates, that you can position this drug also early on, also based on the fact that once obviously a patient has responded or got into remission with cobitolimod, that this seems to be a long lasting effect when considering the data up to 12 months, only after 2 applications.
All right. We are...
Yes,
one quick question then, or a quick answer at least.
This will be a tough one. I want to come to basics. What is it that induces this inflammation? Is this an overreaction to bacteria in the gut, or is it something else? We are looking at ways of influencing the immune response. But unless we knew what induces the problems in the first place, it will be quite difficult.
Yeah. Maybe I can give you my answer so far. So first of all, we have to acknowledge the fact that it's a heterogeneous disease. It's not one disease, it's a number of diseases. And there is one very prominent hit among all those genetic susceptibilities for ulcerative colitis, which is in the HLA locus. So the locus of the histocompatibility complex. So the way on how the immune system is presenting antigens and how these antigens are recognized by the adaptive immune system. So it somehow has an there is an overreactivity of the immune system to something. We don't know what it exactly is, but it could be something autoimmunogenic, it could be bacterial antigens, it could be viral antigens, anyway, anything. Something is here to which there is an overreaction. at least in a substantial proportion of those patients, not covering all of those.
Okay. Well, thank you very much for this first Q&A session with just the doctors, and we'll have a second one at the end of the presentations. Now we have a short coffee break for the people in Stockholm, and people following via the web will need to have their coffee break on their own. And we'll be back at 20 minutes to 11, 10 minutes to 11. So 10:50 we'll start the presentations again. All right. Welcome back to the second half of the Capital Market InDex Pharmaceuticals inaugural Capital Markets Day. Next in line is our Chief Medical Officer, Professor Thomas Knittel, a gastroenterologist from Germany, who will introduce you to the phase 2b study conduct.
Yeah, thank you, Peter. Also, good morning and a warm welcome from my side. I'm Thomas Knittel. I'm Chief Medical Officer at InDex Pharmaceuticals. I joined InDex in 2012, just a couple of days after the first patient was randomized into the COLLECT study. And the COLLECT study, Professor Atrea described you in detail. The primary endpoint was not reached. That was quite a disappointment for us. But we reached several predefined secondary endpoints. And this slide you have already seen. After a single dose of cobitolimod given during endoscopy, 4 weeks later, something more than 30% of the patients were reported remission, so no blood in stool and a heavily reduced stool frequency. When we do endoscopies, 4 weeks, you see mucosal healing with also up to 35% of the patients with a delta of 16% to placebo. And if these two outcome measures, success measures are combined, these two endpoints, to an endpoint which is now the endpoint which is standard in the industry, we see a delta of 18.7%, which is statistically significant in the cobitolimod-treated patients. So this was one study, the COLLECT study. The latest study, the largest study we have done, and Professor Atrea showed you that InDex over the last decade has done a number of other studies which provided similar results. So this observation we have made in COLLECT with, depending on the endpoint, how you define it, with a 15% delta between placebo and cobitolimod, that was also seen in other studies, like in this what we call proof of concept study, started in 2006 with a 13.6% delta, or an earlier study in which different doses of cobitolimod were actually tested some years ago, which, when we take the 30 milligram dose, also showed a 10% delta between placebo-treated and cobitolimod-treated patients. And you see also one problem in this study, that was the pretty high placebo rate. So the data or the observation we made in COLLECT with really statistical significance, this was not a one-timer, but that was also observed in the other studies. And if we combine the data of these studies, the patients which received one time 30 milligrams, and look at the endpoint at 4 weeks, we end up in a meta-analysis of a delta of around 12% between placebo-treated and cobitolimod-treated patients. I mentioned this study done quite some time ago, in which cobitolimod was given at different doses as an enema. 2 patients also with moderate to severely active ulcerative colitis. And you see that at doses of 0.3 and 3 milligram, there is not really much a difference to the placebo. It's even a little bit lower, but the remission rate. But if we increase the dose, we see quite a difference between placebo and the patients which receive 30 milligrams and 100 milligrams. And you also see a little bit a dose dependency. So this was one observation we made in the past with patients in clinical studies. We also know from data obtained in the lab, in vitro data, in which we studied human cells which were exposed to cobitolimod at different doses, and then studied what you also already have seen, the anti-inflammatory cytokine IL-10, the expression of IL-10, that we see a dose-dependent increase of IL-10 expression in these human cells. So taken together, in clinical studies, there's the hint that higher doses result in better efficacy, and also in vitro studies, we see a clear dose dependency. So keep this in mind. What we have also observed in all these studies is a pretty good safety profile. As Professor Atreya told you, up to date, 249 patients were exposed to cobitolimod. In all these studies, in human clinical trials, there was actually no safety signal compared to placebo. So it seems to be a pretty safe drug from all the data we have in humans. Before you can go into a human, you of course have to test your drug in animals. So you do toxicological studies. And we have performed a large variety of studies in rodents, so that means rats and mice and also monkeys, which were exposed to cobitolimod via different routes. We received it rectally, we received it as an IV intravenous bolus, and also it was injected under the skin, so subcutaneously, at doses which were pretty high, 100 milligrams per kilogram body weight. So just keep in mind, in Collect we gave 30 milligrams to a human in total. So at much higher doses, we have seen in these animal experiments also no really side effects. The maximum tolerated dose was never reached. And the only stuff we observed was a mild inflammation at the injection site when it was injected subcutaneously, or a marginal increase in the spleen weight, but also these effects were reversible. So from the tox data, it looks like a very safe compound, and the tox data basically drove the dosing in the conduct study. So the doses we now use in the conduct study are backed by the data we have here in these animal experiments. So I've shown you that even with a relatively low dose and even with an infrequent dosing, given one time or maximum 2 times in the Collect study, we receive data with a really competitive efficacy. We have an excellent safety profile in using our clinical data and also our tox package. And this means there is room to increase both the dose or also the dosing frequency. And it's still a matter of debate what is more important now, to increase the dose or the dose frequency. This is something we cannot answer today, but will be answered in the conduct study. And this drove us, after the Collect study, to perform a dose optimization study to check whether higher doses of cobitolimod can increase the already relatively good efficacy even more. So this is now the conduct study. So it's a randomized dose optimization study to evaluate the efficacy and of course safety of cobitolimod in patients with moderate to severe active ulcerative colitis. So in the same patient population we have studied before. Patient population is not changed. And our primary objective is to evaluate the efficacy of cobitolimod given at different doses or at different dose frequencies. The design of this conduct study was heavily driven by the results we have obtained in the former study, the Collect study. As you've seen in the Collect study, we used an endpoint which was based on a scoring system like the chi-score. And we made this unpleasant experience that this was maybe not the best scoring system, and moved back to the Mayo score, which was interestingly used in all other studies of InDex before. So we moved back to our old scoring system and to the scoring system which is now basically standard in the industry. You have also seen that the definition of the endpoint and the measurement of the endpoint is pretty important in these trials. In Collect, we measured the endpoint at week 12. with not spectacular results. We had much better results when we measured the endpoint earlier. And therefore, in the CONDUCT study, we moved away from measuring the endpoint at week 12 to measuring the endpoint at week 6. So we measure earlier, and we have a better definition of the endpoint. For endpoint measurement and also for inclusion of the patients into the clinical trial, we perform a central reading of the endoscopy. This means patient gets an endoscopy. This is taped, and then by a second independent investigator is judged whether the on-site examination or judgment of the disease activity, of the amount of inflammation you see in the endoscopy, really fits to our inclusion criterion. This is standard now. It's requested by the authorities. So you have to do, if you have do trials in the field of IBD in UC, you have to have a central red endoscopy. As I told you, in this trial, we increased the dose and dosing frequency of cobitolimod, and I explained a little bit the rationale behind that. Regarding steroids, in the COLLECT study, the steroids were mandatory, and some people even said when we presented the data about the primary endpoint at week 12, you have done, because of this high placebo rate, the best steroid trial ever done. To avoid this high placebo rate, and also since we do not have clear-cut scientific evidence that steroids are really needed, in the CONDUCT study we allow steroids, but we do not make them mandatory, and thereby we basically follow the inclusion criteria of the other trials which are right now done in the field. Another change in the CONDUCT study compared to the COLLECT study is that we move back to an enema. The COLLECT study, as Professor Atrea said, in this case, cobitolimod was administered during endoscopy with a spray catheter. Now, if you want to increase the dosing frequency, and if you're considering how practical this can be done, an endoscopy to administer the drug, let's say 4 times, that's simply not possible. It's a quite there are nicer procedures around when doing colonoscopies. And also in the trials before, we always used an enema. So basically in CONDUCT, cobitolimod is dissolved in water, and then we have a 50 ml solution which is put in a bottle like this, and using this bottle, cobitolimod is administered via the rectum. So move back to the enema. And the final change, in the COLLECT study, we had patients with left-sided disease, but also with pancolitis. We discussed the design of the CONDUCT study, of course, with the authorities, and it was one request of the authorities to limit our patient population, because we use the enema, to a left-sided patient population. With an enema, you can cover the colon up to the splenic flexure, so the left side of the colon. And there was the thinking on the side of the authorities that your drug should be exposed to the inflamed area, and the area which is inflamed and not exposed to the drug, like in a pancolitis, we had problems to understand that it could have an effect. So they asked us to limit that to the left-sided disease, which is approximately 70% of the population. Endpoint, Professor Reinisch stressed it already. Endpoint definition is pretty important when you do a clinical trial. So how you define your endpoint and how you measure your endpoint is crucial for the success or failure of a clinical trial. And we have an endpoint which is measured at week 6, which exactly follows the guidelines of EMA and FDA of today, which means patients should have no rectal bleeding anymore, we should have a stool frequency in this Mayo score of 0 or 1. 0 means this is a stool frequency a patient has in remission, and 1 means 1 or 2 more stools when in remission. And we should have an endoscopic score of 0 or 1. 0 is normal mucosa and 1 is with very, very mild inflammation. And this is what EMA and FDA ask for. What type of patients are then included into the trial? Basically the same patient population we had in COLLECT. I mean, if you design trials and you have data from one trial, of course you do some changes, but you also have to be aware not to do too many changes. So we stick to the same patient population, which is patients with moderate to severe disease, defined by this Mayo score. And these patients have an active disease despite a conventional treatment. So we have received 5-ASA and have active disease. We have received or are under steroids or had steroids in the past and still have active disease. And we have received at least one immunomodulator and still have active disease. An immunomodulator means either the classical immunomodulators or the biologicals, so antibodies directed against TNF or anti-integrins. And these immunomodulators are not allowed in the trial. In the trial, as a concomitant medication, as a standard of care medication, it's just 5-ASA and steroids and one immunomodulator, which is azathioprine, and this is exactly what we allowed in COLLECT. So, and now my final slide. How does this trial look like? Here you see the study design. In total, we want to include 215 patients into the trial, which are randomized into 5 arms, 1 placebo arm. So these are patients which receive an enema, but just with water, 4 times. Then we test corbitolimod at 3 different doses, at 30 milligrams, which is the COLLECT dose. And then we increase the dose to 125 and 250 milligrams, given twice. Also in the COLLECT study, it was given twice, 1, 2. And then we have a total dose of 500 milligram, and this is the maximum dose we can give based on our tox data we have. So here, increase of the dose, and here we, in the 4th arm, we increase the dose frequency. So instead of giving it 2 times 250 milligrams, we give 4 times 125 milligrams. Primary endpoint a lot earlier than in COLLECT at week 6. And I mean, we have data that already after 1 dose, after 4 weeks, you see nice effects. So we are pretty confident that week 6 is a good time point to look at. And these patients will be followed for another 4 weeks. This was a little bit also a request by EMA, not for the primary endpoint measurement, but we will follow them up for 4 weeks, whether we still, if we are at week 6 in remission, if we are fine at week 6, if this holds on for another 4 weeks. So this is the trial design. Trial is running. And now we will hear more about the execution of this trial.
Thank you very much, Thomas. So then I would like to invite Pernilla Sandvall, our Chief Operating Officer, to the stage while I open her slides here. And Pernilla will give you some insights on how this is actually done, not only on paper. So, please.
Thank you very much. I'm happy to do so. And Thomas, you said that you joined InDex a couple of days after the first patient in COLLECT was dosed, and I joined InDex a month after you. So I've been on the COLLECT journey as well, and I'm very happy to be on the CONDUCT journey now. So in the study, we will recruit 215 patients. And this will be done in 90 clinics in 12 different countries, European countries. So we have Czech Republic and France, we have Germany, Hungary, Italy, Poland, we have Romania, Russia, we have Serbia, Spain, Sweden, and Ukraine. I think I got them all. We also have a so-called cleared IND by the FDA in US. So we could have conduct in US, and we can do future studies in US, but for the conduct we choose not to do that. 90 clinics, it's a big project. For that we need a CRO, a clinical CRO, and a CRO is a company that has the full machinery to do a clinical study. They have all the services and knowledge. They miss one thing though. They don't have a substance. They don't have a research compound that they can develop. So they have the other services. So as then a development company, you outsource to this CRO to actually perform the study. And we have chosen Parexel as our partner. They are globally, they are a big player. And last year they were actually ranked top 5 in the world. They know what they are doing, absolutely. They have almost 19,000 employees in 51 countries. And one of the main reasons why we choose them, I've highlighted here. So they have experience from 30, more actually than 30 studies within ulcerative colitis and Crohn's disease. And even better, they are considered a leader when it comes to this central reading, as you heard of now. And they have their own team, but they do accept external experts. And we're very happy that you, Professor Reinisch, wanted to be part of that team as well. Really give good insight and ensures we have a high quality in this important measurement. They have subsidiaries in all the countries where we do the conduct study, which means that they give a local knowledge to the countries because there are variations. They also know the local language, which is helpful and very important when you communicate with the clinics, with the physicians and nurses, when you correspond with the health authorities and the ethical committees and so on. This process to select a CRO take a little bit of time. We actually started that process prior to the IPO and had selected Parexel, but signed the contract after the IPO, and it was fully signed in January 2017. So they have subsidiaries in all our countries, and one of the main roles that they have in those subsidiaries is the CRA, Clinical Research Associates. They are monitor the clinical study. They are overseeing them. So they ensure that the compliance to the protocol of the study. And the protocol in clinical development is a very confidential, huge document, a large document that explains exactly what you should do in the study, what measurements should be taken and when they should be done, so that everyone does it the same way. And this is really what CRAs are looking after as well. And they visit the clinics, they look at verification of data, they talk about how you recruit patients, they ensure that the adverse events or potential side effects are properly documented and reported, and they communicate frequently with the clinics. There are many questions that arise along the way. So they are really our extended arms to the clinic. And they assure also that the rights, the safety, and the well-being of patients in a study is really protected. We need to remember that this is patients. Of course, this is the responsibility of the treating physicians and nurses, and all our responsible doing clinical development, but this is something that they need to ensure is correct. And this picture is from one of our CRA meetings, where we also had Professor Atreya here to the very left attending. So, as any big project you would have, you just don't give it away to, or you can give it to a vendor and then you can see what happens. But I would not recommend that. And if you even build a house or something, you know that you need to be involved. And there are many studies out there. And it's a competitive landscape. And we need to ensure that we are on the radar of the clinic and of the CRO. So we are a very active sponsor. So this means that we, InDex, we aim to visit most of those 90 clinics, the majority of them. And last week, I want to say Karin Arneson. In the very back there, we have our clinical trial manager. Particularly, Karin is traveling around to our clinics. Last week, you were in Russia. You actually visited 6 clinics at this time. I was last week in Spain, visited 2 clinics. And Peter, you are our really good at French, so you have been to French sites, and of course then Thomas, being our German speaking, you're focusing more on the German sites, but we're really focusing on all sites. In a study, you always have investigator meetings, and in particular, the kick-off meeting is really important. That sets the stage. And now I've done quite a few investigator meetings. I've been to many over the years, and they tend to blur together. It's like they are in Frankfurt or Warsaw or Prague or something, Central Europe place, and you're in a fancy hotel somewhere, and you don't really remember afterwards where you've been or why. So we decided this kickoff meeting should be very special. So we invited all of the clinics to Stockholm. We had the meeting in the Aula Medica at the Karolinska Institute's campus. We had the dinner later on. Oh, that is the Aula. That's where the lecture for the Nobel Prize in Medicine are held. So that, possesses certain environment that was appreciated. We had the dinner at the Vasa Museum, which is also spectacular if you haven't been there before. When you said the welcoming words, we were all on the stage. This is the entire InDex company. We're all here. We answer any questions. We're very engaged. And we played ABBA music in the buses, and we really tried to make it special. And now as we are visiting the clinics, I'll tell you it was a special meeting. And you're nodding slightly here. We also have CRA meetings. So I said CRAs are key players when you do a study. However, they are often home-based because they travel a lot to the hospitals. And you have 1, 2, or 3 CRAs per country. So they are quite alone in the work. Of course, they have a line manager in the subsidiary office of Parexel, but they're quite alone and they don't know each other. They definitely do not know the CRAs in the other countries. So we decided to have a couple of CRA meetings. This is for them to share best practice amongst each other, because there are some CRAs that are really, they have it in the fingertips and they can share and get good ideas to the others as well. But it's also for us to have an opportunity to ensure that they are fully informed, they understand what conduct is, they can answer questions, because they have other studies as well. So this is not, it's not only our study. Why should they pay particular attention to our study, really? So therefore, we want to meet and ensure that they are engaged, they are also interested when they hear about the study. And if time constraints or calendar conflicts, they should prioritize conduct. That's the aim of this whole exercise. And prior to Christmas, we had such a meeting outside Arlanda in a very charming place, and it was super heavy snow that day when they arrived, and we were like, oh my God, but it turned out to be so beautiful on the countryside and with Christmas trees. And the people working at that place, they had arranged a Lucia-tåg with the kids in this size, and we had the julbord. So apart from being a good meeting, content-wise as well, I think they will remember this place as well. We collaborate closely with Parexel on all levels. So Karin, you have weekly meetings with the project manager at Parexel. You have daily contact with Parexel. We have a governance committee, And such a committee, then we have two prominent members. It's the Vice President of Clinical Operations in Europe, Parexel, and a Senior Director of the Northern European countries for client services. We meet every second, third month, something like that. And we have our chairman of the board has met one of the senior VPs in the executive management team, and they will meet again in May. And I want to say, to us, this is a big project, a big study. To Parexel and my history at Merck, it's one of their studies. So I must say that we have really got attention, being such a small company, and we're really on the Parexel radar, I would say. And then we have Professor Atreya and Reinisch. We are very happy that you are so active in the study and accessible to us, because there are questions nowadays and we get your advice. It's not just the design of the study, but it's also during the study that this is very important and very helpful. So thank you. So we are in a competitive landscape. We need to brand the study. So we have this. You might have seen that on a few slides. I think it's a little, an N is now, you know what it is. It's a colon and it's more inflamed on the left side. And we highlight in UC in the world. And we have this material. And the material is for patient education, for them to understand what's the study about and be better informed. It can also be used as posters. It is material for clinicians, nurses, with details, inclusion, exclusion criteria, a card to have in your pocket, everything like that to help. And it's up to the clinic how to use it. Everything that you show a patient has to be approved by the ethical committee in that country or even at that hospital. It varies. And then we can change the word and local language to patients, always. I want to emphasize that we do internet and social recruitment, patient recruitment as well, in those countries where it's accepted nowadays. This is a developing, to me it's a given, it will come in all countries, but now it's more prominent in the Western countries and Germany, France, Italy being best places, but also Poland and Hungary are good countries for this. Use the same material, we have a website, but how would a patient find a website? So then you have to do ads on social media, Facebook for instance, or a banner at a UC portal where patients frequently visit. They click on the ad, they get to the study website, and then they can answer question and see if they fit the study. Eventually, they will then talk to a nurse in a local language. If they're still interested, if it still seems like the study is a good match, the clinic will be informed, and then they can contact the patients. This is a big project, of course. I've used it many, many times. We used it in Collect as well. And it's not only that patients get to the clinics, but it also that it makes a noise in the field and with the clinics who are reminded about the study. And every word approved by the ECs in that part as well. So if you are a patient, a short flavor what it could be, you get an informed consent, which means that you are fully informed about the study and you need to sign a form. And then you go through screening and randomization and to ensure you fit the study. Randomization, what treatment? You fulfill e-diaries with your symptoms, and you visit the clinic for those dosing that Thomas showed you. And then the primary efficacy is at week 6 and then week 10. This is more described in the annual report. You can take a deeper look there if you want to. So then, this slide. Where are we now? We had the first patient enrolled in June last year, and this was with Professor Atreya and his team. Thank you very much. The study is now approved in 11 countries. Romania is not yet approved, totally silence. We are not the only study not approved in Romania. Something is happening here. And, oh, it's hard to see on the screen. Something is missing here. But Romania is not a big player in the study, so we'll see. 4 sites. it has not a huge impact. Recently, Italy was approved a little later than we thought. We will have all Italian sites up and running by the end of April or maybe early May, something like that. And we have had the first patient in Italy. So that is really good that they are coming aboard. And the patient recruitment is developing as expected. And we have no safety concern, which is very important, as we heard before. We have a data safety monitoring committee, 2 external gastroenterologists, not in this room, we don't have contact with them. They get data for safety, and we have a biostatistician, and they meet regularly. And if no findings, they say, proceed as planned, and this is what they said so far. And the objective is then still to report the top-line results end of this year. You see the country disposition here, and we plan to have 30% of the sites in the Western Europe, 30% in the Eastern Europe, Central Europe, and then 40% of the sites in the Eastern Europe. That's the plan. One last slide I want to show. This is how the recruitment looked in COLLECT, the prior study. So the disposition for this study was that we have 7, or really 6, contributing countries, where we had 38 clinics, and if you do the math, they had 3.4 patients per clinic in that study. We aimed for 120 patients, and we had 131. And this is how the recruitment, so in 15 months we recruited the study, the blue line being screen patients and the orange one randomized patients. And you see it's not like linear or S-shaped or something, and this is summertime and this is Christmas, and it's not like any plateaus. This was really a good exponential curve, which can be explained that as more sites come along, they start to recruit patients, because it's not like every site is approved the same day in a study, because there are health authorities and EC approvals that can take quite a long time in some countries. And as you have had 1 patient, it's actually easier to take the second, then you know all the procedures. So I'm actually very proud to show you this curve from the COLLECT study, and we worked hard for it, and we are working as hard for the CONDUCT study, and my intention is to show similar recruitment curve later on. Thank you.
Thank you very much, Pernilla. And we are running a little bit late, but I will still like to say a few, well, not a few words, quite a lot of words on the commercial potential of cobitolimod. And then after this, we'll have a question and answer session again with all the speakers. So as Professor Reini showed in his presentation, the current treatment paradigm is that you start with 5-ASA or aminosalicylates. There are many out there. It's an old compound, a lot of generics. And if you can't control the disease with that, you have to introduce steroids, preferably a short course of steroids, because the side effects of steroids are during long-term treatment are not nice. So you should not get stuck on that. But some patients do. Either they stop responding to steroids, or they need to be on steroids not to get into a flare. Then we get to what used to be called the third line, but since it's getting quite crowded, it's probably an obsolete expression. But let's say the moderate to severe patients that have not responded to the easier treatments, where we have the anti-TNF-alphas and the anti-integrins as biologics, and the old immunomodulators like azathioprine, for example. And that's where we intend to position cobitolimod somewhere. And the question was debated earlier exactly where, and I guess that remains to be seen in detail. And if you can't control the disease with drugs, you end up in colectomy, and Professor Reinisch clearly described how that is not really a cure. So if we focus on the moderate to severe or the third line treatments, we have the biologics, and we've also heard the shortcomings of the biologics, so fairly limited efficacy. A lot of patients don't get helped by these drugs. The ones that do respond initially might develop tolerance. About 50% of the TNF-alpha responders develop tolerance over the course of a year or two, so they stop responding and you need to rotate them to another brand or another class of drugs. And they are have some severe side effects. So despite well, I should maybe mention the delta, which you've also seen that in an induction, so the initial short course of treatment, we see deltas of around 10% for the currently approved drugs, which means there is 10% more patients in remission in the active arm versus the placebo arm, which means there are quite a few patients that are not in remission. So despite these shortcomings, biologics sell for more than $5 billion per year. That's primarily driven by infliximab and adalimumab, which are more than 2 billion each, around 2 billion each. And now we have a fast uptake curve for vedolizumab, which is closing in on $1 billion sales in ulcerative colitis alone, $2 billion in IBD altogether. So about 200,000 patients globally receive treatment with biologics, and they're quite expensive. Still the average cost of $20,000 per patient and year, and we see the biosimilars of anti-TNF-alpha are coming in around 65% of the originator price. We've also seen the quite busy development pipeline in the industry with a lot of potential new drugs under development. So far, nobody's really cracked the efficacy issue, and I'm sure we can hear more about the scientific reasons for that later. But typically, we've seen deltas of between 10 and 20% in the phase 2 or phase 3 studies of things that are on the way towards the market, and several of them have also increased risk of severe side effects. So to give an overview of what's in the late stage pipeline now, a bit more detailed than what Professor Reini showed. A lot of drugs, but not that many new classes of drugs. So we have the anti-integrins, so those are, we already have Entyvio on the market, and there are a number of other anti-integrins on the way, with etrolizumab from Roche or SHP647, originally developed by Pfizer, now owned by Shire. We have the JAK inhibitors with tofacitinib from Pfizer, which will most likely be approved this year. And then you have a whole range of drugs following that. The JAK inhibitors already approved in rheumatoid arthritis, for example, and a lot of these drugs are developed in several diseases at the same time, hoping to repeat the success of the TNF-alpha inhibitors. Then we have the S1P1 receptor modulators. Ozanimod from Celgene is in phase 3. Had a 10% delta in phase 2. They're also used in MS, for example. Ozanimod had done phase 3 studies in MS. We have the anti-IL-12, or rather anti-IL-23, with ustekinumab or Stelara from Janssen, which is approved for psoriasis and some other things. And we have both Lilly and AbbVie have thrown in their anti-IL-23 antibodies in the race here. Professor Reinisch talked about apremilast, the PDE4 inhibitor, also known as Otezla, approved in psoriasis or psoriasis arthritis. And then there's cobitolimod from InDex Pharmaceuticals. And if you look at the deltas here, they've now been converted to the full Mayo score to make them comparable. So it's the same endpoints in all of these studies, which might not have been the primary endpoint of the study, but to make the relative comparison, you see that it's somewhere between 10 and 20%, and cobitolimod coming in the middle, somewhere around 15%. More on cobitolimod then. Why do we think it's a good idea to develop to spend money on the development of this drug? Well, again, a novel and unique mechanism of action. We're the only one that has come this far in IBD with this mechanism of action, and there is nobody really close to us. As you saw for the other classes, there are a number of compounds for each class under development. We have achieved clinical proof of concept, as Professor Atreya and later Thomas showed, with a competitive efficacy and a very nice safety profile. It's a local treatment that can provide a rapid onset of action. Also probably explains the safety profile because we don't get any systemic exposure really. And now we're running this dose optimization study, hoping to get a better efficacy than what we've seen before, which would also be better than other drugs under development. Maintaining the excellent safety profile, that's the hope. And so far it looks good, as Pernilla said. So we see that cobitolimod has a high market potential as a safer and more efficacious alternative to biologics. And if we look at vedolizumab, which was the first new mechanism of action for 10 years or so when it came a couple of years back, they're closing in on $1 billion in sales in ulcerative colitis alone. So we see that cobitolimod could have a blockbuster potential here. We've heard what the professors would like to see from new drugs. Here's a slide borrowed from another key opinion leader in Europe called Gert Hens from 2014, when he spoke what he would like to see from new drugs in this field. Number one, a different mechanism of action, and if we apply this then to cobitolimod, we meet that criteria. Better rates of inducing clinical remission. Yes, so far, so good. Less loss of response in responders, and that's really the issue of tolerance. And as we don't have any systemic exposure, we don't see any theoretical reason why why patients would develop tolerance to this drug. Efficacy in primary and secondary non-responders to anti-TNF. Professor Atreya showed that data and underscored the importance of it. Evidence of efficacy and safety in combination therapy. We've had a lot of the patients in our trials have been on a lot of other drugs at the same time, not TNF-alpha though, or other biologics, but on azathioprine, for example, steroids, 5-ASA. Less immunogenicity. That's again comes with the lack of systemic exposure. Less risk of infection, that it's not really an immunosuppressant, but more of an immunomodulator. Again, no immunosuppression. And without systemic exposure, it shouldn't reach the fetus if we have any pregnant patients. So the actual product, the chemical, the drug substance, it's a synthetically manufactured oligonucleotide. So it's not really a small molecule, but then again, it's not a big biologic either, a bulky protein. So it allows a less complex manufacturing process than we have for the biologics, which means we can have a competitive cost of goods. The cost of making this would be lower per dose than for a biologic. So synthetically manufactured also allows a better control of the manufacturing process. And then it's a simple formulation in an aqueous solution. As Thomas said, the final product is 50 milliliters delivered with an enema. The intended storage condition is 5 degrees, so refrigerator. And there we have a current shelf life of 36 months. So it's a stable product. And given then as a 50 ml solution to the site of inflammation. In the conduct study, the physician or the nurse actually delivers the drug. But there is definitely potential that the patient can do this themselves. As we've heard, patients typically start on rectal 5-ASA. Their disease journey starts with rectal 5-ASA. So they're used to taking rectal products. Still, it would be nice to have an oral formulation. I mean, a pill a day is kind of a standard medical treatment. So we've started to look into the potential of making an oral formulation which would deliver the active substance to the colon. So it's not a systemic, but a topical treatment still, but you take it via the oral route rather than the rectal route. And that would be for life cycle management purposes. So it's not like the launch product would be the rectal, but then as a second generation product, we could have an oral, and it would allow us to access certain patient populations better, like pediatrics or children, for example. And there are also some markets, like for example in the United States, where they prefer pills over rectal enemas. However, remember that the most sold drugs today are injectables. Infliximab is an intravenous infusion. You need to go to an infusion center. Or adalimumab is a subcutaneous injection that you can do yourself. Still injections. We did a fairly ambitious market research not so long ago, where we had an expert consultancy firm in the US go out and interview doctors and patients in the US and in the 5 big European countries, a total of 148 patient responses via a web survey and 65 physician interviews via telephone. About 40% of this sample was from the United States. The objective was really to understand better what sort of patients do these doctors have, how are they treated, to hear their reactions to our target product profile. So what do they think of the product as we think it will be like, and to understand their views on the proposed formulation and mode of administration, really drilling down into this question of rectal mode of administration. The overall response was very positive from both physicians and patients, and features such as fast onset of action, efficacy and tolerability scored very highly. And the message was really, if this works, we will use it, in simple terms. Of course, patents or IP is very important in this business if you are going to be successful. For cobitolimod, we've built a strong IP position. It's based on a broad portfolio of use patents. The composition of matter has expired, but we have we have actually 5 patent families, but 3 key patent families that protect the use of cobitolimod to treat inflammatory diseases. The first one is granted in the US, Europe, and Japan, and it expires in 2026 or 2027, and then you have a 5-year patent term extension possibility, so it would take us beyond 2030. We have the second family, which is also approved in Australia and Canada. Same time frame, would cover us beyond 2030 with the patent term extension. And then more recently, we've had the third patent family started to get granted. It's been approved in the US and in Japan. We're waiting for Europe and Canada. That would expire in 2032, plus the 5 years would take us to 2037. We are continuously looking at new patent opportunities based on our old studies and ongoing and future studies. So we have a quite active IP activity. And in addition to patents, cobitolimod will be subject to the data protection from regulatory agencies, meaning nobody will be allowed to launch a copy in a country, 10 years from market approval in Europe, 5 years from the market approval in the US, and 8 years from market approval in Japan as an extra protection. So that brings us to our business development effort. So the aim is really to find a partner for cobitolimod prior to phase 3. Somebody large enough and skilled enough to provide the resources and expertise for the phase 3, the last stage of development, the registration with the authorities, and then the launch and commercialization of the product. Typically, this is done via licenses, where the small biotech then licenses the drug to a bigger pharma, and in return gets upfront payment, milestones, and royalties on a percentage on the future sales. And we are, in parallel with the conduct study, working on our package, our business development package, where the aim is really to have a compound which is ready to start phase 3 in 2019. So there are some toxicology work we're doing. We're doing some work on the manufacturing in parallel to the conduct study. And we have a very active business development effort. I spend a lot of my time on meeting pharma companies, explaining what we do, getting them interested in the product. We have a senior business development consultant named Andrew Thompson, who is supporting us. We have Charlotte Admire, who is our BD manager, and also Thomas Knittel, our CMO, is actively involved in these meetings. And we participate at the main partnering conferences. There are a couple of big conferences each year where the big pharmaceutical companies go and meet smaller companies, and they meet each other as well to do deals. And we usually go to the US and Europe, and we've also been to Japan, to the Bio Japan. So that's really more commercial or deal discussions. Then we have the important scientific conferences. We've heard about the ECHO conference. There is the DDW in the US, UEGW in Europe, where we've been very successful at having presentations, posters. Professor Atreya has presented several times at ECHO, and now more recently we had this mechanism of action data at ECHO, and we will have a poster also at DDW, or two posters at DDW in the US in May. The feedback we get is that there is a clear demand from industry for promising assets in this within IBD. So really, despite all the activity there is ongoing, there are many companies actively looking for additional assets. And we've established good contact with potential partners. We know who we should talk to within the therapeutic areas of gastroenterology and inflammation or autoimmunity. I think we're well positioned there. So what does the pharma industry think these sort of assets are worth? And the deals that's been done in this space really underscores the significant unmet medical need and therefore the commercial opportunity here. And we've seen a string of really high profile deals. Celgene has been the most aggressive. They started in 2014 by licensing Mongersen, which unfortunately later on failed in phase 3 in Crohn's. But they paid $710 million up front for an asset which at the time had phase 2 data in Crohn's only. The same Celgene actually bought all of Receptos, a California biotech that had ozanimod, which is now in phase 3, with a 10% delta in ulcerative colitis. They paid $7.2 billion. Admittedly, ozanimod is also very promising drug in multiple sclerosis, but even if you take a fraction of the $7 billion and point that to the ulcerative colitis, it's a lot of money. Then more recently, we have Shire's deal with Pfizer, where they licensed an anti-integrin, which is an IBD product, can't be used in anything else. Paid $90 million up front, $500 million in milestones. And in February this year, J&J licensed a phase 1 asset from Theravance, which is a gut-restricted JAK inhibitor, and paid $100 million up front. They promised to pay another $200 million at phase 2 readout, and then $700 more million of milestones plus royalty and profit sharing. So this really highlights that IBD is a high-value indication, and there is a lot of interest from industry. So again, let me round this off with the strengths of InDex and cobitolimod. Large commercial potential. Ulcerative colitis is a difficult disease we've heard with high unmet medical need. The current products or biologics particularly sell for more than $5 billion a year despite their shortcomings, and cobitolimod therefore has a high market potential as an alternative with a novel mechanism of action. we've come a long way. We have a lot of clinical experience with cobitolimod with a good safety profile. The main results we are expecting end of this year, where we have the potential to get a substantially higher efficacy than we've seen before, and which would make it a very attractive asset for potential partners ahead of phase 3. It would also validate our other DIMS assets with the potential in inflammation, securing the next step of the company. And we have our experienced board and management supported by a very dedicated team of professionals. So that rounds off the actual presentations, and we will now have a Q&A session with all the speakers on stage, and we should get a table up here as well. Anna tar bordet. Microphones on.
Yes, now they are.
On. So you go. I'll be on the corner here next to Pernilla.
Good. Is it on? Yeah.
Are we all on? Maybe we should dive into questions from the audience in the room here, if there are any. So Matthias, maybe?
Yeah, I guess I can start. This is mostly related to Professor Atrea's presentation, I think, about the COLLECT study and what you learned from that. I think, the main question, I guess, is how predictive are these results? And since there's been some changes in both trial design and mode of administration, maybe we can talk a little bit about that.
Yeah. I think that's a very valid question that we also asked ourselves when we were to design the new CONDUCT study. What can we take out of it and how valid is it? I think if you really ask me regarding the design of the COLLECT study, I was even surprised that there are positive signals, because the design was really not in favor of the compound with a 2-time application and then looking after 12 weeks. So if you look at week 4, after just 1 application, which is not much, you see all the drugs that we are using at the moment, I think within 4 weeks they are used daily or at least 2 applications with the antibodies that we have. Just seeing there a positive signal shows you there is a potential. And then of course we asked ourselves, especially with a local application, if you increase frequency and dosing, I think you would at least expect probably even higher efficacy. And I think one of the main problems that you'd like to avoid in this phase 3 trial is that there's underdosing, because I think this is a general problem in our field, that probably we are not utilizing the whole potential of the drugs. So I think from the design of the study, the timing, the frequency, dosing, and looking at endpoints, I think it's very valid. And I think based on the positive signals that we see in phase 2 with just 1 application, I would expect that we should see positive signals with this trial design.
Okay. And then you have 5 arms, right, with 215 patients. How some treatment failures will obviously impact the results. What do you think about this?
The question of the potential of high placebo rate.
Yes.
Would you like to address that, Thomas? Or Professor Reinisch, why do we?
Yes, I can touch upon this, of course. The point is, I think Professor Atrea has noticed this before, that one major, that there were several reasons why the placebo response rate or remission rate in the COLLECT study was very high. It was a noisy endpoint measurement. It was a late time point in the assessment of week 12, which always, so to speak, supports higher placebo response rates based on concomitant treatment with corticosteroids. And what was not so much stressed maybe before is the fact that there was no central reading in place. And now with CONDACT we have an endpoint at week 6 where there is supposed to be a less clear impact of concomitant steroids, and we have central reading in place from which we do know that central reading is clearly reducing any background noise and therefore also the placebo rate. So my expectation would be that with this design, the placebo monster, so to speak, should be well tamed.
I can
Thanks.
maybe expand on that. The study is really designed to show a delta of 20% or more. We will also catch a delta lower than that, but we're actually swinging for a bit higher delta than the 10% that people have had. So we've kind of raised the bar there a bit.
In terms of the business development, if you have the hypothesis that you will have a good study, what is it that you have most problems with explaining for the pharmas, the big pharmas that you're meeting? What is your sort of big task in that, in getting them to understand that this is something good?
Well, of course, they would like to see a positive study, and with the COLLECT study, even though we see these clear signals or clear efficacy in the secondary endpoints, which are also the more relevant endpoints, it is always a problem if you haven't met the primary endpoint of your study. So that's one of the most important objectives of the CONDUCT study, is to get that confirmation. Yeah. So and apart from that, I think, and it's we have our mechanism of action, and if you look at the other classes under development, they are very well mapped in the sense that there are so many assets from so many big pharmas that they really know these pathways. We're alone with ours. We're building a lot, but about the mechanistic data. So that's why I think it was so key with the data Professor Atrea's lab have now generated, where we can really show how this anti-inflammatory effect that follows from the treatment of cobitolimod and where we are in the inflammatory pathways. I think that's
maybe to add. Big Pharma is definitely interested in this compound, and our problem in the past was that we had not enough clinical data really with a p-value. If you talk to Big Pharma, their thinking is quite simple. If you have not, as Peter said, reached the primary endpoint, this is a negative study, and then you are in an explaining mode, even if you have met several secondary endpoints and have a nice mechanism of action data, they do not really hear so closely to you. And the reaction of Big Pharma right now is, well, interesting compound, interesting mechanism of action. Please come back when you have data from CONDUCT. So I'm pretty optimistic when we have met the primary endpoint in the CONDUCT study, that we are in a completely different league talking to Big Pharma as we were in with the COLLECT data.
Lars?
Yes, I'd like to come back to the increase of the dosage in the CONDUCT study. Could you talk a little bit about what you expect in terms of efficiency and what you've seen in the data before? Or expect, rather hope for, I guess.
In the clinical study we have done years ago, where 30 and 100 milligram were tested, we see a little bit of tendency that a 100 milligram dose is maybe or could be more effective than the 30 milligram dose. With the 30 milligram dose, we are in a, depending on how you define your endpoint, you are in a 15 plus minus range. So my personal expectation is that increasing the dose, or maybe the dose frequency, should lift us to 20% plus. This is, but I mean, right now we don't have really data, hard data to support this. But also from the animal experiments we have done, from the in vitro stuff we have done, we have seen this dose dependency, and it would be stupid not to test a higher dose. I mean, the safety profile is nice. It allows higher dosing, and we could come up in a completely different league. And that would be, in my eyes, also a little bit a game changer in the UC field.
Well, can I then twist that question and ask the more independent speakers here? What kind of efficacy or delta would you like to see for a new drug to be effective?
Yeah, if I may start to elaborate on this, I think what we are currently used to see is, as you anyhow indicated before, is a difference of 10 to 12%. So anything which would go beyond this signal of 10 to 12% would be already more than even promising, I would say. And with regard to the dose dependency, we should also bear in mind that in a natural system, of course, it makes sense to test higher doses, but it's not a guarantee that higher doses are also achieving better outcomes. It depends upon the saturation of the receptor structures and so on and so forth. And we have seen this with anti-adhesion molecules, where not necessarily higher doses with an anti-adhesion molecule leads to better efficacy. And therefore it's also important to learn more about this biological system of TLR9s, to ramp up the dose and to see how far can we go, what is the saturation of the receptor most likely look like in the human system. And this would give us much more insight about the potential in the end of the drug and how to position it.
Maybe to add something, of course we also talk to TLR9 experts about their feeling about increasing the dose and dose frequency. And I remember one statement of really someone really skilled in the art. He said, well, the only possibility for you is to test it in a clinical trial in the human setting. All the stuff you have done before, this is some evidence, some hints, but at the end of the day, you have to go into a human and test it. And this is what we are actually doing.
And yeah, just to add to the points already mentioned, I think if you look at efficacy, you should also look at safety, because we are still in this, let's say, 10 to 20% field, but none of the drug has gone beyond that. Of course, having a drug then reaching 20% would be great. But I think if you look at the oncological field, what are they doing to achieve higher efficacy is they are combining drugs. So I think this is something that we are also slowly entering, but I think a big prerequisite to combine drugs is that they have to be safe. Because we cannot combine two antibody classes, because we already know just using them as monotherapy, they already have a quite considerable adverse event potential. So of course combining then these antibodies from safety reasons, I think it would be very delicate. So even, of course, this is looking into the future. If you probably have to achieve even higher levels, you'll combine drugs, but you need safe drugs. You can only combine them if they are safe. So I think then if you have good efficacy and safety, and then of course you have the potential perhaps by combining them with other drugs to even achieve higher levels. But of course this is looking into the future, but I think this is something that we have to do to reach higher levels.
We do have a question from the internet here. What about the prospect of dietary or other non-medicine treatment for ulcerative colitis? It would be great for patients, but as investigators, this might be seen as investor, this might be seen as a risk in the future. Yeah. So what if you can just change your diet and then we don't need any drugs?
I think there is a lot of research activity currently ongoing with regard to enteral nutrition, and not only enteral nutrition, but also with modifying diets in patients with particularly Crohn's disease, less UC. So UC is in these kind of trials always under appreciated or underrepresented. And in Crohn's disease, there are some signals in favor of this. But the question, what is the product to be sold as such. If I simply change the diet and could find out that the change in the diet does make has major impact on the efficacy. And once again, as Crohn's disease and ulcerative colitis are very heterogeneous diseases, it might easily be the case that diet changes in the diets might have some good effects in some patients, but in others it's completely worthless. And I myself, I tried it via different small concepts during my time in Canada, and I need to say, even putting the patients on a gluten-free diet, what they are sometimes doing anyhow, I couldn't find any major effects on short term, either on the biomarkers or clinically. But I also need to admit that there are some approaches which try to foster, so to speak, the epithelial barrier. And those principles seem to fly, and they are highly appealing to patients. And there is a great hype around fecal microbiota transplantation, and most likely the mode of action which we're talking here about, cobitolimod, maybe plays around some similar principles as fecal microbiota transplantation is touching upon. Based on the dysbiosis, there might be less signaling through the TLR9, and maybe by signaling via TLR9 you could get the balance in the homeostasis, right? We don't know, it's very hypothetical. But nevertheless, there is a big interest from patients, and each week I receive an email from a patient, are you up and with a study on fecal microbiota transplantation? And we did some studies, and we did some meta-analysis, and actually showed that that there is some efficacy, but I don't think it's the future. Because it's only a principle to show that if you modulate, so to speak, not necessarily directly the adaptive or the innate immune system, you can also achieve something very meaningful in those diseases by modulating, for example, the microbiota composition. And this is such a big learning that you don't need to suppress with an efficacious drug always the immune system to get the balance right. There might be other ways, like the ways which are discussed here, to do this job correctly.
And maybe it's also the patient population, because if you these diets, probiotics are tested, but mainly in patients with mild disease. And here we test cobitolimod in patients with really moderate to severe disease, and you have seen how these colon mucosa looks like if we have a grading at 2 and more. So I have some doubts whether a diet can really cure the patient in a severe disease. But I would not exclude that maybe in a mild disease it has some beneficial effects.
I think we have to round up, unless there's one last question then from Matthias.
Thanks. So I was thinking about back to the positioning and the market acceptance of cobitolimod, for example. How, as biologics are quite established by now, how would you guys consider the potential for cobitolimod in the market? Where would doctors use it? How would they think? Are they conservative or open to try this before biologics and such?
Perhaps I may start. I think biologics, yes, they are accepted, but also we don't have any alternatives. So we know all the downsides that we have with biologics, but what should we do else? So I think what we are really looking at is, again, of course, I'm rounding this up, is efficacy and safety. And safety is something that is troubling for us with biologics, and also loss of response, immunogenicity. So these are all the topics that are hotly discussed. How can we circumvent them with biologics? But as they are systemic therapies, there's no way really to get around it. So we have to take them into account in our therapy decisions and try to make the best of it. So I think that really reflects the unmet need at the moment that is still there. And also, although there are many developments, we have seen this with the JAK inhibitors, but again, they're associated with side effects that I think we'll even encounter new side effects that we are not aware of at the moment. So we have to be ready for that. So I think the search is really ongoing, because with all these new substances, there's still the need for something else. And again, it's really efficacy and safety. So if this is shown, of course, in the study and then in the hopefully subsequent phase 3 trial, I think there'll without doubt be a position for this drug, because it really fulfills the criteria that you pose for every new drug that likes to enter the market.
All right, that would have to be the closing words. We've run over our time schedule a bit here. I'd like to thank everybody in the room and everybody who followed the webcast or who will view the replay on our website eventually. And now I would like to invite the guests here in Stockholm to the buffet outside, where we will have the speakers, but also other InDex representatives that you can talk to and continue the discussion. Professor Satreya, Professor Reinisch, thank you very much for coming, and it was a very we learned a lot, and thank you so much.